very important experiment is transferring to millions of language as a textbook example. I learned this as long as I'm 12 years old in the junior middle school in China. And so many years, it's become a puzzle of all the exam papers, of the young scientist competition, all tells us what it means. It's a very typical example, experiment that tells us the genetic information. Okay, so what happened? This is experiment is done, but the work has to do. 94 days. Everybody is busy. Is that right? The world is not good. What happened? World War. Not good life at the time, but people still keep doing something. We want to see what is this genetic information for? What it is? It is DNA, it is RNA or protein. Luckily, in 1944, there is a three very intelligent scientists in the United States. Avery, McCarty, and McLeod. Usually this paper is three of them are co-first author. In Rockefeller Institution, Research Institution in NYC. Right now it's not Rockefeller, at that time it's a Rockefeller University. Right now it's a Rockefeller Institution of Medical Center in NYC. In 1944, they published a paper and they did a very smart research to verify it is DNA, it is not RNA. How they did it? Okay, I have this tube, which has a rough Streptococcus ammonia. Okay, I'm gonna grab after I cultivate it. I'm gonna use the agar medium. This agar medium used to cultivate smooth Streptococcus ammonia. So I planted there. Is there a colony? No, of course no colony. So this is a negative control. So I want to verify. These things happen here. Is some genetic chemical there. So what we do? I usually use another color to express it. Okay, I have using another tube. This tube is a smooth Streptococcus ammonia extract. And then I combine it with another tube. This tube is rough one. Okay, I'm going to add it here. Then using same agar media, which is smooth Streptococcus ammonia media. And I find a colony there at the beginning, which is good, same thing. Genetic information is transferred. Okay, how about that? Let's add some of the enzymes there. I add DNase. What means DNase? The DNase could hydrolyze DNA. Then what happened? This agar is nothing grow. Because the DNA is hydrolyzed. Okay, I'm going to add RNase. What means RNase? Hydrolyzed RNA. However, DNA is there, and the protein is there, and other chemicals there. This agar is growing. Growing smooth Streptococcus ammonia. Okay, I want to verify it is not protein. So I add proteins. Look at these all enzymes. Again, what you see. This agar is growing. The smooth Streptococcus ammonia is there. What this tells us? This tells us this genetic information transferring in Griffith's study, very possible it is DNA. Because others are gone. Now here is something we want to mention. What is the advantage compared to this study to this study? Not only for they verified, use a smart experimental set, said it is DNA. This is a big one from live animal research to agar median. This saves a lot. If you keep doing this, there's millions of mice gonna die there. This use agar median, it's a breakthrough because we're doing the tubes. We save lots of the energy, lots of the time, and also lots of the cost. So it is a breakthrough. Okay, now we have this DNA figured out. We have this DNA figured out. That's okay, that's a study right here. Only one experiment tells you it is DNA. That is not enough. Okay, we have to do something. Is that right? We need to make sure it is really DNA, not others. So what happened? 1952. Hershey-Chase experiment. Okay, you know something happened in 1953. We get to that real quick. Not real quick today, next week. 1952. This is 1944. It's already 8 years later. Okay, everybody recovered from the Second World War, still making progress. This is not a candy bar. Okay, Alfred Hershey, not Hershey chocolate. This is not Chase Bank, Mercer Chase. At the time, there was a research associate in, I believe it's in Stanford University of Caltech, some area there. Alfred Hershey is still there, maybe almost 100 years old, pretty much. Later on, he's a professor in University of Oregon, in Eugene, Oregon. You know Eugene, Oregon, all the athletes there is for the competing to go to the Olympic Games. Eugene, Oregon, okay? Hershey-Chase experiment. What they did is they are using bacteriophage. These studies, when you think about it, is very interesting. Now, why they use bacteriophage? How come they suddenly jump and use bacteriophage? By the time this Spanish flu is still going, but it's because it accumulated, so all we can say mutation comes out, so it's not that strong. Then people want to find some of the therapy, we call it a phage therapy. We're using these days, using in the veterinary science area a lot, which means drinking what? Virus. Phage is a virus. So what it looks like? This is a phage. If you know this is a bacteriophage, what it looks like? Usually it's not like this. It's actually pentagon like that. Phage is a virus. This virus, you're going to eat in bacteria. You could eat a bacteria. Okay? We call it a lytic phage. You also could inject the genome sequencing, which is DNA, into the bacterial chromosome. We call it a warm, friendly, or temperate phage. And at this moment, he uses a T2 phage. It is a very warm phage, a very friendly phage. What are the major components for the virus? It's so simple if you know about these days. On the top, this is capsid. The major component is protein. And what in the middle of the center? This is genetics, or genetic chemical, or DNA, maybe RNA, or we say, but never both. Okay, we'll talk about that. And this T2 phage, lots of the pre-experiment did already. It's possibly, most likely, is DNA. So he did that. He prepared. Now the thing is that you have a bacteria. Okay, this is the bacterial genome. So a phage is there. Now of course this is like a tail, somebody tail, it's a template, like this is a collar, some people say it. These are the other things. Then when we go there, this genome will be integrated here. This DNA gonna be there. Okay, so how are we gonna do? I'm gonna label something. Is that right? So what I label it? I use isotope. Isotope. Isotopes has already happened because the nuclear weapon is developed. So isotopes is being widely used by the time. Okay, now here is something I want to ask you. What is the major chemical in protein? However, it's not existing in DNA. Some people say nitrite, is that right? Because nitrite is a major chemical in protein. But it's not, DNA has it. Adenine has it. So what is that? Sulfate. Isotope sulfate, 35. What is going to be the major chemical for DNA? Phosphates, is that right? So P, we make it 32. So he ended up with two experiments. I had a bacteriophage right here. This is a bacteriophage. Okay. This bacteriophage, I'm going to label it. How are we going to label? The outside, the capsid or the coat, I'm going to label S35. Then I'm going to find the bacteria. It is a bacteria. The bacteria like this. So very important that this one we're going to go there. Okay, so the phage is there. So DNA will go inside. Okay, then afterwards what he did. By the time there is a centrifuge, I don't think people, when they edited the books, they understand it. They actually know centrifuge. Oh, okay, this is the older version, it's good. The new book, I read it, they put a centrifuge there, that's not good, this is good. By the time there's no centrifuge, so it is difficult to isolate it. Is that right? You have a virus, you have a bacteria, what are we going to do? They worked a very long time. They find a kitchen blender is a good idea, which is to make juice these days. You just put that in, and it's going to spin. It's a good idea then. That's what kitchen blender is like a centrifuge. Because they could separate them. What happened? Precipitation. That's what I said, the panicles or sediments. This is the bacteria. And on the top of the supernatants, this is the phage. And this is the virus. So what they find when they label S35 of the capsid, they find it's very interesting when you come out here. This phage, a new phage, it is actually S35. Of course, there's DNA there. It comes out on the top. On the top, there is all S35. And at the bottom is actually something mixed. Okay, so what this means? This means there's nothing coming for the bacteria because everything is only, the coating is coated on the top. So he did the second one. The second one, what he has is still a phage. And this phage, in the DNA, he's going to label S32. Okay, then you go here, you find a bacteria. Find a bacteria, and this phage go in there. Okay? Then this DNA is going to go in, and then you have this bacterial chromosome, and the DNA is going inside. And then the same thing, I'm doing the kitchen blender. We're going to blend the spin. Okay, what happened? They find this bacteria. bacteria showing up at the bottom has P32. What this means? Which means as long as I label P32 for the DNA, the DNA is transcript, not transformed into a bacterial chromosome, and because when you use a kitchen blender, it spins down the bacteria at the precipitation, you find P32. That means the DNA is transferred, and this mechanism right now, we call it a conjugation. Conjugation is rely on the bacteriophage. Okay? And here, by the way, DNA transformation. There is a transfer, there are three methods. Number one, transformation. DNA to DNA, naked DNA to DNA. Conjugation. You learned in your genetic class. This is rely on bacteriophage. Last one, transduction. Transduction rely on F. phyli. Is that right? That's the three methods. Here, not only we verify the DNA is a genetic information because it's shown in the bacterial cell in the precipitation. We only know this is a method. We also know this is a method called conjugation. So after those three very typical experiments, we almost know DNA is a genetic information. Okay, so next Tuesday we're going to talk about the DNA structure. There's another three important history experiment we have to mention. Then we talk about the DNA replication. Okay.